Hello everyone, my name is Sofia Garrido. I'm an undergrad biology student at Universidad del Rosario in Colombia, and today I'm going to be summarizing some of the key points of the fine seminar by Alexander Ophir about how variation in parental care impact the social brain, social behavior, and reproductive decision making. Dr. Ophir is interested in the individual differences in the neurobiology of social behavior and how this influences making decisions. But before going into detail about his trajectory regarding this interest, I'd like to start by establishing some of the fundamental framework to understand more about his work. Firstly, it's important to define what is meant by decision making, which in this case is an individual's ability to contextually assess a situation and react based on past experiences, as well as the actor's access to information. To study reproductive decision making and identify the sources of variation that influence this process, Dr. Ophir has focused on the prey voles as his study system. They are socially monogamous rodents, which leads to other sets of behaviors, like, for example, biparental care, forming long-term relationships with a single partner, as well as defending their home territories. These characteristics make them a starting model to test the hypothesis establishing that early life social experiences alter developmental trajectories to shape reproductive decision making. Some of the background investigations that allow room for this hypothesis are related to differences in parental care, as well as the plasticity of neural mechanisms. Regarding the first point, and as shown in the figure, it's been found that biparentally raised pups receive more care than uniparentally raised pups, but the source of the reduction comes primarily from the father. Additionally, for low-care parents, fathers tend to give significantly less care. All of these results indicate that a major source of social variability is given by the natal nest and by fathers in particular. On to the second point of neural mechanisms, the main focus are nanopeptides, more specifically vasopressin and oxytocin, which are ancient signaling molecules linked with social behavior, whether it be aggression, affiliation, dominance, communication, and many others. It was found that these nanopeptides are highly sensitive to changes in the environment, especially in the postnatal environment, and in the case of male vasopressin receptor, it's particularly sensitive to environmental perturbations. Hence, could variation in parental care be responsible for generating phenotypic variation in brain behavior relationships observing reproductive decision making? To test this, one of the experiments consisting of housing voles so that they had to walk a tube of around 5 feet 2 if they wanted to access food. For some enclosures, this tube was flat, and in others, it had a 30-degree incline, making it harder to get the food. In this way, a trade-off for the parents, whether single or biparental, was created where they had to choose if they took care of themselves or of their offspring. Digging further, when carrying out a social approach test, it was found that males that were raised in the trade-off condition were significantly slower to approach another conspecific, suggesting that the influence of paternal care impacts male social behavior and potentially also impacts reproductive decision-making. Now, the lateral septum is a brain area, shown in purple in the image, that plays a key role in modulating context-specific motivational responses and states, which is a good starting point to link the previous results with neural phenotypes. Also, it's been shown that when vasopressin binds to its receptor in the lateral septum, it'll decrease social interaction. And so the question of if variation in vasopressin receptors could explain differences in social approach arises. To answer this question, epigenetic modification via DNA methylation was carried out, and it was shown that voles raised under the trade-off condition exhibited more methylation in the 3' UTR than the non-trade-off animals, suggesting that animals raised in the trade-off condition have increased vasopressin receptor in the lateral septum, as indicated by the positive significant correlation between these two variables. Pushing it further, when looking at the percentage of methylation in relation to latency to approach, only males in the trade-off condition show a significant and positive correlation. Hence, epigenetically increased lateral septum vasopressin receptors, attributable to differences in paternal care, affect social approach. But what about the role of the lateral septum in sociability and monogamy in prairie voles? To see this, using DREADS, they activated neurons in the lateral septum for the individuals in the treatment condition and then carried out a series of behavioral tests. In the case of the social approach test, there was no difference in the control groups, but the lateral septum activated individuals spent significantly more time in the social zone. In the case of the reticent and intruder test, it was done both before and after pair bonding to evaluate aggressive interactions, and in this case, controls were more aggressive after pair bonding, but the lateral septum activated individuals stayed non-aggressive and even became more prosocial as they spent more time cuddling with the intruders. Finally, in the partner preference tests, selectivity of a partner over a stranger was maintained in both control and lateral septum activated individuals, which all leads to the conclusion that the lateral septum manipulation affects sociability but not bond strength. However, the search doesn't stop here, as reproductive decision-making in prairie voles is only one of the many examples on how it is possible to link different levels of analysis, for example, ontogeny mechanism and function, in order to understand more about social behavior and cognition. I invite you to go and check out the complete seminar to go more in depth about what I talked about in this video, and thank you so much for watching.